So we um, talk about some the amino acid and size of macromolecules, and the last time we do some lithography. So we today we go a little bit more into some specific topic among the amino acid. We have 20 different amino acid. We want to focus on some special amino acid, which is tyrosine. And the tyrosine is indicated by the Y in a single character system. And at the beginning of this class, I emphasized that you should memorize the amino acid structures. So this is the amino acid structure. Then you can draw either this way or that way, and you have a basically pi ring structure with one hydroxyl group. And also, so you maybe remember your acid base, the concept you learn in general chemistry, which you have a pKa you learn in the aqueous solutions, and depending on a pH, you have deprotonations of this side chain, which with pKa of the for specific tyrosine is about 10, so 9 or 10, depending on the context, which is amino acid sequence of the protein, and also the location of the protein. Either the tyrosine can be located inside or outside, so that the varies the pKa. So the pKa about say 10, and then the physiological pH is about 7.5 or 7.4. So usually this pKa is higher than the physiological pH, so it's protonated. So and then the pH you have remember the Henderson-Hasselbalch equations, that pH equals to pKa plus the log 10 basis accident base, so that the pKa equals to pH when the acid species and protonated species and deprotonated species is equal. So the log on same concentration, log 1 is entirely becomes a 0. So this term is gone. So pKa and pH equals to pH. So when the pH is higher and higher, go about 10, then this protonated and deprotonated species becomes 50-50. So usually at the physiological conditions you have a protonated version. But you will see in the later in this slide that cases are different. Actually, in the real protein chemistry, there's deprotonated tyrosine is actually found in many different systems in protein. So let's go one by one. The first one is resilient. So this is the the terms that resilience. That resilience is kind of some materials terms that difficult to actually tear, right? This kind of resilience, also elastic-like materials. So the resilient is IN means is a protein. So the resilient protein. This is what the names indicated for. So it's like elastic rubber materials. So the resilient is an elastomeric protein found in many insects. Actually, when you think about the insect body, and also there's a wing, and wing means this very fast moving and up and down, and then at the interface between if this is the body, and if this is the wing, then there's an interface between the body and the wing, right? So at that point we have the resilient in many insects, because you can think you have to have some rubber-like materials to have a stable the wing movement in nature of in many insect cases, especially when the insect has to be moved very fast for very, very high frequency of the wing movement. Then actually the resilient property has to be very good. So as you see, it is a part of the what enables insect many jump or pivot their wings efficiently. first discovered by Torkel in the ring hinge. Actually, hinge is very difficult. It's important region in insect. So the chemistry now is unusual chemistry actually. So you can see here the tyrosine. This is the protein backbone, and also this is protein backbone. And then there's a side chain which is the C alpha carbon and CH2 and phenyl rings and OH, which is the tyrosine structure. And there are two tyrosines actually connecting to each other. At the end of this class, I will explain this chemistry a little bit more when I explain the melanin chemistry. Okay, but actually, to have this covalent bond between these two, the phenol group, you have to have some oxidation chemistry involved. That means electrons from the tyrosine released, that becomes O double bonds and C quinone is formed, and this quinone is chemically reactive. And the chemically reactive quinone and quinone here, and then they actually bond together, recovering the electrons from the neighboring phenols, and they form the covalent bond. So this the cross-linking between the protein chain is very, very critical for maintaining this elastic properties of the protein. So this most efficient elastic protein known, and then is tendon. It's a tendon-like protein, and also this 97%. It's only 30% of 3% of stored energy is lost as heat. Okay, so it's not, you can read all the sentences, but this region is a hinge region in this pair. You have the two tyrosines and dityrosine form, and then two electrons is out of it. And then these two, the phenol rings is actually a fluorescence. It's a fluorescence probe. So you can have a fluorescence signal in this resilient area. You can see the blue signals, blue fluorescent signal when you activate it by the UV light. So you can see very, very clearly that the proresilin, and there's a tyrosine is crossing resilin and dityrosine here. You can actually see this, not only this dityrosine, the liquid chromatography, but also you can see by the simple UV providing illuminating here in this hinge region, you can actually see the existence of the dityrosines here. So this you can see it, one protein chains and one protein chains and the other protein chains, and you have to imagine three-dimensionally. The tyrosine is crossing each other, and then also there's a protein. They also connected to each other by these dityrosine formations. Also, not only this one protein has multiple tyrosine, and then this tyrosine, that tyrosine, multiple cross-linking between the proteins. So then these proteins you can actually try to pull this protein, and then they actually resist against your applied force out of this protein materials. So very interesting chemistry you can see here. So the first one is tyrosine is involved with increasing mechanical properties of the protein materials. That is number one cases. You can see the second cases now. The second case is green fluorescence protein. So the green fluorescence protein is the most protein, I should say, in the protein world that has to be using for imaging the inside of the cell or in vivo, where our interest of protein we have, and then we have a protein we tag with often by the GFP that these fused proteins you expressed in vivo directly or the cells, and then you can actually track this protein where the proteins are inside the cell or membranes or Golgi or nucleus, cytosols and lysosomes, whatever. Not only within the cell, but also you can actually monitor the movement of the cell inside the body, so on and so forth. So it's got a Nobel Prize, of course, to study of the discovery of this green fluorescence protein. So now, the most important part is tyrosine in this green fluorescence protein. So the second topic is tyrosine. Tyrosine is involved with illuminating, so it's exhibiting the photoactive properties. Is this you have glycine, tyrosine here, and then we got some steric hindrance, rotations of this tyrosine to have actually this nitrogen. This is the amide bond. This is very interesting chemistry. The amide bond CONH here is not chemically reactive, right? So you can simply imagine that you have protein backbone, the amide bond repeating backbone. So this amide bond is chemically stable. That's why you have alpha helix and beta strand and three-dimensional structures. But in this case of the GFP, this amide bond is actually chemically reactive. It's chemically reactive to react each other, and then these rotations are sterically hindered. So this means that this kinetics from the energetically stable structures to rotate is very slow process, slow step. And then what happens is the reactions is very fast. Okay, so when that happens, and the cyclizations, dehydration, oxidations are all happens very fast. So that is number one. So number one is tyrosine is involved. Okay, so tyrosine is involved, and then these locations now, these locations in here is within this barrel. You can see this is a barrels of it's like a wine barrel. So you can have all the beta strand are wrapping around this tyrosine, glycine, the threonine structures, which is fluorophore here, and then of course, as you learn, inside of the protein they fold it, and then there's no water molecules, H2O, within these proteins, right? So you have a proteins transcription and translations in the ribosome. We have proteins. You learn about this the coronavirus section as well. Proteins is created, and then they fold it forming into three-dimensional structures. Of course, inside of the protein there's no water molecules. Same as here. So there's no water molecules in here, and barrels are formed very stable barrier, not preventing all the H2O molecules go inside. So they keep very hydrophobic environment. This hydrophobic environment actually the lowering this pKa. So I mentioned pKa usually 10 or 9, or lowering much low pKa that actually physiological condition you actually O minus, not OH, is existed in these structures. So it's a little bit details in chemistry that you have now. See this backbone flip. This is a very rarely happening in these backbones of the chemistries, and then also this and then the flip, and then there's the nitrogen attack, and then everything cyclization reactions in here, and also the cyclized reaction is stabilized by the positive and negative charge nearby the amino acid. Okay, so this is the situations occurs in the fluorophore region of the GFP, and also what happens in the researchers in GFP is actually they want to change their photo characters, fluorescence characters, from the green and reds and purples, blue, yellowish, and then you can change the colors by giving the mutations. Now see the wild type phenolic anions, which are phenols and stack pi electrons and stacking. Indole formations, imidazoles in the chromophore also. So totally different approaches. What happens is nearby the chromophore, 65, 64, 65, see the 66, so on and so forth. Okay, like nearby the chromophore, so that happens. So this, you see this, this class 4 is phenolate, which is pi stacking, also pi stacking electron systems giving yellowish fluorescence protein. And then this cyan is the indole formations in providing the cyan fluorescence protein. You got a more blue region, and blue region is the imidazole in the chromophore. Okay, so this is you don't need to remember all this, but there's a you have to know that there's a tyrosine is key to provide this beautiful colors of the fluorescence by this reaction nearby with the amino acid, and also the nearby the mutations of this protein provided varying the colors of the fluorescence from the green to blue, green to cyan to yellow. Okay, so this is the key. The second properties, which is fluorescence properties by the tyrosine. So the number 3 is also the providing that the yellowish, but this is fluorescence is different. This is absorption based. So you see by your eye, you see the color same from this yellow protein or the GFP. You can see color, but as you know, the fluorescence you need the activations, right? Activations of the molecule, and then the molecule become the excited state, and then they become grounded states, and then there's photons released from this photophore. This protein is basically like a color absorption based. So they're absorbing some specific wavelengths of the light, especially in the visible region, which is 400 to 700 nanometers. It's like the same as the amino acid they absorb in UV. DNA is 260, or tyrosine is 280, tryptophan is 280. So it's not providing the photon. They simply absorb the correlated corresponding the wavelength region. Same as here. So this chromophore is paracumaric acid. We called this paracumaric acid means it originally becomes COO minus as an acid, carboxylic group is here. So this enzyme they form the thioester form, and then this is basically looks like a tyrosine residue after reaction with the cysteine 69 in this protein. Basically they provide a yellowish color. And also, as you see, this protein structures is also similar to GFP. They located inside this protein. That means no H2O molecules, and they're very hydrophobic. As I mentioned, this becomes O minus. pKa becomes lower in the hydrophobic region. It's like you can imagine like a lipid, no molecules, and they hating or expelling the H2O out of the environment. So lowering pKa. So now, this O minus has to be existing in the very hydrophobic region. It's like lipid region, which has to be stabilized by the hydrogen bond formation, which is glutamic acid 46 and tyrosine. Another tyrosine here. So they form hydrogen bond donor and acceptor region, and then they become stabilized in there. Also, okay. So then O minus and phenyl ring double bonds and CO. So now this you learn about the conjugated electrons, right? Conjugate electrons, they resonate form. This electrons conjugated double bond, single bond, double bond, single, double, single, double, single, double bond. So it's conjugated structures that are conducting the polymer. So it's conjugated electrons. The electrons are travels freely in this molecule. So they basically lose the energy. And then losing energy means that generally redshifted this tyrosine. This tyrosine region basically 280 nanometers absorption. But they have another conjugate structures they provided becomes lose the energy. Absorption maximum goes to 300 nanometer region. Additional conjugation more. Deprotonation become O minus more. The redshifted, so that becomes 350 400, so this resulting in 446 nanometer absorption, which is a blue absorption. So they absorb blue light. What we see is the complementary color of the blue region, right? So that's why we see the color. So they have all the white light comes, and the blue regions, the molecules are absorbed, and we see the rest of the color in the visible range, and then this is what we complementary color. So we complementary color of the blue is yellow. So then we see this protein. You may recall this PYP, photoactive yellow protein, because we have a specific protein purification classes. You go back to our previous class, the specific example how to purify this protein is this PYP. This is the actual protocol to purify the PYP. Okay. So I will continue with the melanin formation, which is another pigmentation of our skin. Okay.